Um, Hi everyone, I'm Naomi. I'm a data scientist at Sable Bio, and we want to welcome you and thank you all for joining today's webinar. We're really excited to talk to you about how we approach target safety assessment at Sable. So as a quick background for today's webinar, um, anyone who's conducted a target safety assessment will understand the struggle of sourcing and combining all the data sources required to develop a proper understanding of a gene's risk profile. We have to understand the basic biology of the target in question, as well as investigate its preclinical and clinical history. And each of these categories is broad, covers many different data types that live in different databases that vary in quality and accessibility, and not all of which are kept up to date. At Sable, we've built an interactive platform to collect and process these large and disparate data and surface relevant target safety insights for interpretation. To do this, we've developed a risk scoring process that we'll be diving into in greater detail in today's talks. We have 3 talks today, each one highlighting a recent addition to our platform in our latest data and app release. We're lucky to have our CEO, Josh, talk us through Sable's approach to clinical data gathering and processing, followed by our Director of AI, Yuha, who will talk us through his team's literature extraction pipeline. And last but not least, our lead computational biologist, Claire, who's going to discuss how we derive directional risks from biomedical data. Your questions are very welcome throughout. Please use the Q&A tab on the call to ask your questions as and when they come to you. If there are any particularly specific questions, we will address these at the end of each talk, and more general questions we'll save to answer at the end of the webinar. So let's get into it. Josh, if you wouldn't mind.
Thanks, Naomi. Yeah, as mentioned, I'm going to be giving our first talk about grounding emerging clinical assets in real time. We've been building this pipeline for a couple of years now, and yeah, ready to show it off. So with that, clinical data and target safety assessments is obviously a gold standard for validating a target safety and biological profile. If there is in-human data, while it can be convoluted by various patient populations and drug effects, and ultimately is what you're held against when you're starting to fill out IND enabling reports as well as emerging drug therapeutic programs. The issue is when you have an emerging target class, so maybe it's a new field, a new oncological target, the visibility of these most recent assets is it's not a need to have, it's a must to have, it's a competitive necessity. However, when it comes to public registries, there is a delay in reporting. So new clinical activity is often unrecorded for months, if not years. So manual collation, if I was doing a TSA a few years ago, I would go to places like Kemble, I'd go to Google, I would start reading papers, I would go to catalogs, try and find tool compounds. However, what we're going to present today is Sable's clinical pipeline, which is up to date. It runs weekly and it has the freshest data. It's comprehensive. It doesn't miss anything. And ultimately, it's flexible. So you still have complete user control over its predictions. So what are the clinical assets associated with my target? This is the question we're trying to ask. And at Sable, we have a general philosophy of trying to break down and modularize questions to a point where they're less subjective and they're a lot more simple to interpret. And more importantly, the methods that we create are easier to validate. And so in this question, we're actually asking 2 separate things. One is which drugs are in clinical trials. This is simple. This is a lot more simple of a task. Simply, it's a statement of fact. When a trial starts, we should be able to get the drug associated with it out. The second question, which we'll get into a little bit more, is conceptually a little bit more difficult. Which drugs hit my target? And so you see, breaking this down into 2 steps, we're able to potentially get a bit of a higher quality asset identification. But even if we miss one, say in the second question, we're still able to surface all drugs in clinical trials at scale. So starting with the first one, as I mentioned, it's a little bit easier. Which drugs are in clinical trials? We have things like clinical registries. So at Sable, we use clinicaltrials.gov, which gives us very good coverage across the globe. There are gaps in this when it comes to especially the Japanese and Chinese industries with early stage stuff, but anything past early phase 1, typically you'll find in clinicaltrials.gov. This sits in a kind of semi-structured format. So we have the trial ID, we have a description. So in this case, a multiple dose study of PF 046 to, I'm not going to say the whole name, because I'll tongue tie myself, in type 2 diabetes patients. We have three arms. We have a low dose, 2.5 mgs, a higher dose, 5 mgs, and a matching placebo arm. And so we've made a number of specialist models that extract each one of these fields at scale. So now we have a tabularized format. So we know the intervention, this PF04. We know the disease trialed, the treatment arms, lower dose and higher dose, as well as the control. And again, this is something that if you were to look at it, you'd say, I could do this, in a few seconds, maybe, a little bit longer, depending on the complexity of the study. That's the point. And I think these really simple questions are very easy to validate at scale because of it. But we're able to do a lot of really interesting things when we have this data across the entire registry. The second question is a little bit harder, and I think it's worth calling out the fact that external data is just really difficult to handle when it comes to high quality relationships between drugs and targets. You'll have a catalog, it'll usually claim something like a potent and selective inhibitor of X. You then have Kemble, so it'll often show the bioactivity for every target other than X. And then you have a number of publications of varying reproducibility and quality. And again, given a large amount of time, one can put together an idea of whether or not this is a high quality clinical asset or not. The ultimate reality of the assignment is typically if it was really important to you, you would buy the tool, you would make the tool or the clinical asset, and you would test it internally to see um, the true nature, at least with your assay technology, what targets it actually hits. However, without the ability to do that, what we have lent into is the ability to see where multiple external sources align, where we can gain confidence in that initial assessment. So as mentioned, there are bioactivity databases, there are tool catalogs, there are recent news articles, there are regulatory documents, all of which we feed again into a proprietary model that allows us to spit out a few different interesting pieces of data around these assets. These are primary MOAs, so the targets that people intended to hit with these drugs. Secondary MOAs, these might be common off targets, family off targets, essentially targets that people didn't mean to hit with this drug, but do. And more importantly, as well as the confidence. So we like to maximize recall wherever possible, but you should know that if a single paper mentions that it might hit target X, that would be a lower confidence than if all these sources align across the board. The nice thing about this is any prediction we make within this, we have access again to that source data. So you're able to see exactly why that prediction between a drug and a target has been made. So an example of this, we have another Pfizer compound. It's a different one. I don't know why I'm picking on Pfizer. PF04217903. A catalog EPSA mentions that it's a potent and selective c-Met kinase inhibitor with a huge selectivity fold over a number of other kinases. And there's lots of data out there, lots of biomedical bioactivity data in Kemble. However, in the Sable platform, front and center, we highlight that this compound also has significant off-target activity against a number of phosphodiesterases. And this is a kind of target class that you wouldn't necessarily consider screening a kinase inhibitor against. And interestingly enough, there was a paper a few years after its publication where they found that it did a number of strange things in rats, and it took a long time to figure out the cause of that, and it was in fact this off-target activity. And again, to call out, this is something that many chemists, scientists on the call could eventually get to the same conclusion. However, we've pre-baked this information from the very beginning to save you the time and effort it would take to read these supplementary info and various papers attached to this compound. Another really good example is T2. This is a really emerging oncology class coming out of a number of different companies. If you were to go to Kemble, for example, there would be no clinical assets registered to this particular target or many of its family members. However, within the Sable Target Intelligence platform, we have 7 early stage phase 1, phase 2 pan-TEAD inhibitors that act via the YAP-TAZ pathway. And again, I think for these emerging target classes where arguably there is the most competitive need to be in the know, we have a huge window of opportunity to improve on these public registries from the very beginning. And kind of even with these assignments and going back to the original call of it's never perfect and we're not going to get it right 100% of the time. And arguably, even if all the data aligns and then you test it internally, you might find that um, actually it's a pan kinase or it's a pan PDE or something like that. Within the reports that you make in our platform, you're able to add drugs that you think we've missed via a free text search bar. And more importantly, you're able to remove drugs that you think actually this target safety assessment is flooded with risks. It's all due to, in this case, papaverine, which is a natural product. I simply want to remove it and any risks associated with it. And so ultimately, you're still in control of your report and the quality of them. So hopefully, during this talk, I've kind of given you a whistle-stop tour of how we ground emerging clinical assets in real time. These pipelines run every week, so your reports are always up to date. And you can access the data today if you're already a customer of the target safety assessments via our 2.0 release. And if you are more data centric, potentially a data scientist, you can access via our clinical API as well to integrate into your own workflows, whether or not they're ML models or agentic or anything in between. And with that, that's the end of my talk.
Thank you very much, Josh. I think we have one question that's specific enough to ask now, which is how do you do quality control on all of these drug and target predictions?
So quality control at Sable is a really interesting thing that we have a general philosophy about, and we use the concept of human in the loop methodology. So we might run this method 100, 200 times, and ultimately we have scientists within the organization, in this case, myself. I will look through the results, I will sanity check what is good, what is bad, what isn't working quite well. And usually that iterative loop lasts a few months of multiple cycles of really trying to improve it to a point where if we were to sample a large number of predictions across the board, if any of them are wrong, it doesn't get released to customers. So overall, we're really happy with it. It takes a little bit more time, but I think it's worth the extra effort.
Thank you very much. If anyone has any clarifying questions or follow-up questions, then please feel free to ask them, and we can tackle them at the end of the webinar. And now, if we can navigate the swap over of screen share, we'll pass over to Yuha to talk about literature at scale. Thank you.
You are on mute. It's okay. We were going to have a technical hiccup at some point. You can tell as a company, we're not Zoom users.
All right. I really want to stick with literature pipelines. All right, so I'm sharing, am I?
Yeah,
Yes,
you're all good.
you are.
Very good. Okay, so let's start again. So, hello, my talk is about our recent work to extract safety relevant target adverse event relationships at scale. Uh, we cover generally quite a lot of different safety related structured data sets. But the unstructured data from scientific literature is providing additional increase in the coverage. In this talk, I will walk through the motivation for the work, details on how we model the problem, and finally show you how that impacts the users in Sable application. So starting from some facts on literature, so it's really hard to keep up to date what's coming through the PubMed Central and of course the commercial publishers. There's so many documents coming in every day. So here on the graph on the right, you can see the annual publication numbers in PubMed. So started from about 750K in 2006. Of course, there are earlier documents as well, but this is just the last 20 years. But last year we had almost 2 million documents, and this year already over 1 million in just a little bit more than 4 months. You can see that the amount of literature is really increasing almost exponentially. So it's really hard to keep up to date what's going on. For us, literature is particularly important for the emerging targets that don't yet have that much information in the clinical side. So we aim to extract information that's then combined with the other data modalities that you use is going to give you some additional information. In the company, we have extensive experience in working with scientific literature and building scalable data pipelines as well. So we hope that this work is something that will be useful for our customers and will take the Sable application to the next level. The goal of the work in general was to make sure that we have literature-wide relationships available in the application, and we do frequent updates. In practical terms, this means that we are updating the data once a week. So once a week, we get a fresh set of data, and we provide that to the applications. And as we are a relatively young organization, we are still building our foundations for ML modeling, and we wanted to make sure that we apply industry best practices so that we use fine-tuning of domain-aware models, scalable data engineering practices, and model tracking as examples. We also wanted to apply other model training approaches like active learning, which allows us to focus on the things that are hard for the models and make sure that we can, with less effort, make better models in the end. And with any machine learning approach, benchmark is hugely important. So we have hand-curated benchmark datasets for all the models we have to ensure that we can quantify the performance and know that what type of improvements are making sense for us to apply. Next, I'm going to talk a little bit more about the way how we're modeling the data, and then I'll move on more to the actual modeling approach that we have. So you quite often when you read scientific articles, you may face a paragraph like this. So it's a really long paragraph, has a lot of information of this particular target, TREM2, also talks about quite a few other things. And we would like to know what is its relationship with Alzheimer's disease. And a single paragraph like this contains so much information. It's really hard for humans. It's really hard for computers to understand and make a summary and consistent predictions that what is the essential association or relationship between the target and the phenotype in this case. So you could also go to the other extreme, which is like, well, let's process sentences. Sentences are great because they're relatively simple, easy to read, but they don't have that much context information, so they don't hold necessary information that sometimes is required to make a good prediction for the relationship. Therefore, we found a middle ground, which we call the snippets. And a snippet is a rolling window of sentences that captures the bigger picture of what's happening within the paragraph, but still making it quite easy or easier to digest by the users and by our machine learning algorithms. Here you can see an example from the same paragraph where it's been split into smaller pieces, and you can see that TREM2 is repeating a lot, and you can also see the Alzheimer's disease here as well, mentioned quite a few times. And now it starts to be a little bit easier, at least for me, and I hope that most of our users agree with me that this is something that makes a little bit more sense in terms of a representation level for the data. It also ensures that any information we show in the application is kind of more consistently of similar length in the snippets. Okay, so data is modeled in a sort of a semi-paragraph level, which allows us to ensure that we can run predictions efficiently, and the models are utilizing their context in a more appropriate way. Now I'm going to focus a little bit on the modeling problems. So the question that we really want to answer is that what is the modulation on a target? Is there an effect on the adverse event or phenotype or disease that we're looking at? And is there an association or relationship between those two? Is there a strong relationship or very, we could say, a mild association type of relationship? And we split this into three distinct models that are taking care of their own individual problem. As an example, for the target modulation model, we're looking at the target age stack 6 in this paragraph, and we can clearly see that there is an increase in that gene or protein in this text. The next thing is that we want to know what's happening with the adverse events, and we can see that there's an ischemia, and again there is an increase, and we can conclude with the second model that we're looking at a potential increase of ischemia caused by something. And the final model then looks at the language to see that whether there is a relationship between the two. And in this case, we can say that there's at least an association. It's not necessarily very strong, but at least you can say that, well, there is some evidence somewhere about it. And this is the way how we process the data, and then in the end we combine all this information into a single observation or single prediction that then is taken further into the application. All right, so having looked at the models, I'm going to have a few more slides about the pipelines. So in high level, the literature pipeline works, and you can probably find a million different implementations of scientific literature pipelines in all over the world. But essentially, you have your articles. Very commonly apply entity recognition. You're trying to find the biological concepts you're interested in, the targets, the adverse events, cell type, species, whatever you think that are going to make your data more rich. And after that, we apply the models. So we have the models here, the target modulation, the adverse event modulation, and the relationship model. And after that, the information is combined into what we call evidence. And these are individual relationships that hold a number of the source snippets that I talked about earlier. And eventually this data is then taken into the application database, which is then serving the application with the data on demand. And we have about 5 million of these relationships from the scientific literature using this modeling approach. Pipelines in practice are quite a bit more complicated. So I think currently we have up to 100 individual processing steps that make this whole system, starting from the deliverables from PubMed, for example, and on the other end will be data placed into the database. So this all happens automated once a week, and we get an updated data set. All right. So what's the effect of all this work? So as I said earlier, we have data from different modalities like genetics, we have the clinical data, and now we're adding information from scientific literature. So from this picture, you can see that scientific literature has like an overwhelmingly more data coming into the application compared to the other data sources, which kind of makes sense because clinical trials, the frequency of them isn't very high, but obviously a lot of articles and a lot of research is happening in parallel all over the world. And we think that this is very important. And as you can see, that these are only the sort of directional associations where we know that a specific modulation has a very concrete effect on an adverse event. There are more of those more general associations where the direction isn't as clear. Okay, as a user of Sable platform, what does this mean to you? So essentially, this works as part of the reporting system. We have all the evidence that we extract from scientific literature is available to you in the platform. Furthermore, you can go very deep into the data if you wish to do so, looking at the individual phenotypes, the individual relationships. You can do searches additionally if you want to. But essentially, what we're providing you is a sort of a full access to scientific literature that has been pre-digested for your use case. And this is something that you can access through the application, but we also have an API that allows you to access this same data that you see here on the screen. In the beginning, I mentioned the benchmark evaluation. So as I said, we have manually created benchmarks for target modulation, phenotype effects, and the relationship prediction. And on the right-hand side, you can see a confusion matrix for the relationship model, and you can see that the model is doing pretty good job in finding the correct classification for the samples in the benchmark. I should note also that we apply additional thresholding so that we actually only take the most confident predictions out of the system, which will reduce the recall a little bit, but make sure that the precision is very high. As a conclusion, our scientific article processing pipeline is computing corpus-wide relationship predictions, and these are updated once a week. So our users always have most up-to-date scientific literature in their hands. We have just released this data in the version 2 of our application, and you should be able to find it in the application if you are already our users. And as normal, we are working on a number of improvements to the training data model architectures and the confidence measures to make sure that the data is right, and we do everything we can to make it better. If there are any questions, I should be happy to answer.
Thank you, Yiha. I think we have a couple of questions that are probably specific enough to answer now rather than at the end of the webinar. The first is, are you able to separate statements of primary research versus citations in your model?
That's a great question. At the moment, we don't make a separation of that. We always provide the references and the original citations or original snippets from the prediction we made. So you can either follow the trace there, but I think that that's a fair point, and it is hopefully one of the things that we could improve in the future.
Thank you. And then I think the other most specific question would be, how do you deal with the separation of genes and adverse events that have overlapping synonyms? I don't know if it
Ah.
means whether or not genes and adverse events have the same name or multiple genes
Yeah,
and
yeah,
multiple
yeah.
adverse
That's a very specific question. Yeah. Okay. One of those that I really like. Uh, so the models are separate. So we have a separate model that's assessing how the target modulation works and how the phenotype affects. So those models are able to categorize the correctness of that NER output as well. So if we had a phenotype that overlaps in synonym with a gene, let's say, and we can find both when we run our named entity recognition. We find both the target and the phenotype, and then it's up to this model that we have for detecting the target modulation, the phenotype effects, to make a decision which one is the more appropriate. So ideally, in that situation, one of them will say that, well, this is not a valid phenotype, and the other one says, yes, this is a target, and it's got a modulation, or the other way around. But these are statistical models, so they are not perfect either.
Understood. Thank you very much. Okay, so I think we'll move on to Claire's talk now. Claire, our computational biology lead, is based in Tokyo. So rather than force her to give a talk live at midnight local time, she has pre-recorded her final talk, which we will play for you now. And please continue to ask questions throughout. The rest of the team should be able to answer them for you. Josh, that is not coming through for me. I think maybe because you're muted.
Hi everyone. So you've heard today already how we extract information from clinical data and from literature data. So to finish off the webinar today, I'm going to talk about how we combine this and other kinds of biomedical data to produce the prioritized list of potential safety risks that we surface in our target safety report. So the challenge that we face, this is one I'm sure many of you will be familiar with, is that when assessing the potential risk of modulating a target, we're interested in evidence that's very fragmented across lots of different data domains, different databases, different sources, and these are formatted in different ways, and they have to be assessed in different ways as well. So for example, we might be looking at GWAS associations, we might be looking at publications, we might be looking at clinical trials. And these are all very different types of data. Directionality is also very crucial. We want to know specifically what are the risks if I activate or inhibit my gene, not just a list of any phenotype that is associated with the target. This can be difficult to resolve, and this is partly because not all of these data types can provide that directional information. Sometimes we don't know the direction of effect, but we can also have data that has conflicting directions of effect. So for example, we might have publications that point to both an activation risk and an inhibition risk. This can be quite difficult to resolve, even manually. Combining all of this evidence and making a decision can be a challenge. but we want to do this at scale for tens of thousands of potential risks when we're assessing a target. We want to do this systematically. So today I'm going to talk about how we approach this at Sable. So our approach is to divide this up into two parts of the problem. Firstly, we reshape data from various different domains into a common format that we call evidence. And in this step, we're asking, what does each data source tell us about how this target relates to this phenotype? Once we have all of the data in that format, we can then aggregate the evidence together, weigh them up against each other, and infer what's the most likely effect on this phenotype if I activate or inhibit this gene. So I'm going to talk about each of these in turn. And just a note on the terminology here, I'm using the word phenotype. By this I mean any kind of phenotype, adverse event, trait, or disease that can inform us on a potential risk that we might see from modulating this target. So let's make some evidence from clinical data. So in this case, the target of interest is DGAT1, which is a target with known GI issues when inhibited. And so I'm looking at these clinical trial records for drugs that target DGAT1. And when I'm creating this evidence, what I'm asking is, what is happening to the target? What's happening to DGAT1? What's happening to the phenotype? What's the overall relationship between the two? And how strongly does this evidence support that relationship? So in this case, my target is DGAT1. It's being inhibited by these drugs. And nausea is my phenotype. I can see that nausea is occurring in a good chunk of the participants in the clinical trial. So the overall relationship we see here is that inhibition of DGAT1 leads to an increase in nausea. So this is what the clinical evidence looks like. So the gene, the relationship, inhibition increases, and the phenotype. We also have this score, this confidence score on how strong is this evidence. So this is a domain-specific score. So for each different kinds of data, we'll calculate this in a different way because it depends on what kind of data we're looking at. But overall, a score closer to 1 means that the evidence more strongly supports the relationship, and a score closer to 0 means that the evidence more weakly supports the relationship. So for example, for clinical data, we might derive this score based on how many subjects do we see this side effect occurring in? Is it enriched in the treatment versus the control? Do we see this side effect more generally across multiple clinical trials in patients with this indication? And how sure are we that this is linked to specifically DGAT1 inhibition as opposed to drug-related side effects? So these kinds of domain-specific contributors are taken into account when calculating the overall confidence for this evidence. But what we get here is this consistent structure that we can extract from different kinds of data sources. So for example, literature relationships, we might have 3 publications that describe how inhibition of this gene leads to some phenotype X. We would model that as well as gene inhibition increases X. For a GWAS association, we might find gene variants are associated with an increased risk of phenotype X. Now in this case, we might not know the direction of effect on the gene. So for example, we know that this variant is associated with the increased risk, but we don't know whether the variant has an upregulating or a downregulating effect on the gene, so we can't say the overall directionality. In this case, we would model this as gene modulation increases phenotype. So we know that some change in the gene has resulted in an increase in the phenotype, but we don't know which direction it went in, so we wouldn't be able to tell specifically whether this is an inhibition or activation risk, but we think it is a potential risk if we modulate this target. And finally, we might have differential expression evidence. So in this case, we are looking at a tissue sample from patients with the disease, and we see that the gene is upregulated in these tissues. In this case, we don't know whether the upregulated gene expression is causing the disease or is a result of the disease. So in that case, we can only say that there's an association, some kind of correlation between the two. So we know that there's a link, but we don't know whether it's caused by the change in the gene. And so in this way, we can capture the different level of directionality that we can extract, where possible, from the data, and we put them in this common format with this confidence score. So next up, how do we aggregate those together? So what we want to know is, across all of the evidence, across all of the different data domains, what might happen if I activate or inhibit this target based on all of that evidence. And there are three important components that we want to calculate here. We want to know the weight of the evidence. How strongly is the evidence supporting this potential risk? What's the direction? So is this a risk for an inhibitor or an activator? And finally, how consistent is the evidence? So is all of the evidence pointing to an inhibition risk? Or is there a mix? Is some of it pointing to inhibition or activation? So how sure are we about that directionality? And these are the three main things that we're trying to quantify from all of this input data. So on the right here, I have these three bars that I want to fill up based on the evidence. I want to know to what extent is this phenotype a risk if I inhibit my target, if I activate this target, or if I modulate this target. And modulation risk here means if I do any change to my target, will I see a change in the phenotype? So I'm going to give you a quick overview to illustrate how the evidence from different domains is combined to fill up these bars. So for example, let's start with the experimental data. So here we saw that there was an association based on the differential gene expression between my gene and the phenotype. So we don't know whether this is an activation or an inhibition risk. It might not be a risk at all because we don't know which direction this is going in. But this gives us some small chance that there's a modulation risk. It might be. We know there's an association, so it might be the case that if I modulate this gene, there will be a change in the phenotype. Here's some stronger evidence. We have genetics evidence. So we have two different types here. The first is a GWAS study, where we don't know the effect that the variant has on the gene. So I don't know which direction this is pointing to. And I also have a pathogenic variant, which is a loss of function mutation that causes the disease. So in this case, inhibition is increasing my phenotype. So in this case, we combine these both. The modulation increases case contributes to the modulation risk, and with some probability, it contributes to activation and inhibition risk as well. There's a chance that it might be an activation or an inhibition risk because we don't know the direction. The pathogenic variant, however, we know the direction here. It's a loss of function mutation. So this contributes to the modulation risk and the inhibition risk. So that combines with the previous experimental data. We can see how this is starting to build up. Next, we have literature evidence. So in this case, I've got some conflicting data. I have some publications that say that inhibiting the gene increases the phenotype. I've got a good strong signal for that, high. On the other hand, some of the publications with lower confidence report that activation of this target increases the phenotype. So this is in the other direction. So in this case, the literature contributes to the modulation risk because we know some change in the target is causing a change in the phenotype. And we have a small contribution to the activation risk from this lower confidence evidence and a larger contribution to the inhibition risk from this inhibition increases evidence. And finally, we have clinical data. So in this example, I have these inhibitor drugs in clinical trials where we observe an increase in this phenotype as a side effect. So this is strong evidence that inhibition of my target increases the phenotype. This will contribute to both the modulation risk and the inhibition risk. So hopefully this visualization gives you a high-level view of how we are able to combine disparate data types with different kinds of directional signal into this single, uniform, transparent confidence score. And the real model, we use a probabilistic model for this. It's a little bit more complicated than the visualizations I've shown here, but the model is mimicking what a real scientist is doing when they're assessing and weighing up these different kinds of evidence. There's three main behaviors that we have encoded in the model that we think are important. The first is that more evidence from the same domain increases confidence. So if we observe this multiple times in genetic studies or clinical trials, this increases the confidence. But we have diminishing returns as this increases, because we don't want any one domain to dominate the prediction. Secondly, and more importantly, evidence from multiple different domains compounds to increase our confidence. So when we have orthogonal signals from, say, clinical and genetics, that's a stronger support than just from genetics or clinical alone. On the other hand, though, we know we don't have a complete view of the data. It might be that, for example, there are no clinical trials for this target, or there are no GWAS studies for this phenotype. So in that case, when we have this lack of evidence or missing in a domain, it doesn't harm the score. It doesn't reduce our confidence. But when we have multiple different types, we have stronger confidence. And finally, hopefully as you've seen from this illustration here, we have evidence with mixed directionality. Sometimes we have directional information, sometimes we don't. But what we're able to do is we capture the most directional information that we can, and all of this contributes when considering the final result. So even if we don't know the complete direction, it's still able to contribute to our final risk scores. So what we have overall is we've gone from different kinds of evidence from different kinds of data, which is very hard to weigh up, to this uniform, transparent confidence score. And not only does this tell us whether we think this is more likely to be an activation or an inhibition risk, it also tells us the level of support that we have. And this allows us to rank across phenotypes. So some phenotypes where this is higher, these will appear higher up in your report, so you're able to see the more likely phenotypes first. So what does this look like in the app? I have here a screenshot from a target safety report for DGAT1, and I'm looking at a specific phenotype here. And I can see at the top here, this visualization shows that there's strong support for risk from inhibition, but there's bidirectional evidence. And this visualization shows what I was showing on the previous slide. So we can see there's more support for this being an inhibition risk than activation risk. This is a summary across all of the evidence. And we can also see this broken down by domain, and this is useful so we can see if the signal is being driven by a particular domain or if two different domains maybe disagree. So in this case, we can see that the clinical data is all pointing to an inhibition risk, whereas the genetics in the literature, they are both a bit more mixed, but they both point to overall an inhibition risk. And experimental, we don't have any evidence in this domain for this phenotype. So this gives us this high-level view. It's taking this messy evidence landscape and giving you this clear signal that this is an inhibition risk, and this is strongly supported. But we also want to keep the detail, because we want to make sure we understand why this is the case. So underneath this, you can see the underlying evidence that goes into this visualization. We can see the clinical trials themselves, the genetic evidence underneath here. So what I'm showing here is for a specific phenotype, but we can also zoom in further to look at the individual evidence in more detail, or we can zoom out and see what are all of the phenotypes that are associated with this target, how do they group together into similar clusters, and which ones are more strongly supported than others. And in that way, we support this multi-level exploration. So we can look at either the high-level risks, high-level summary, what are the main risks, what's the direction, or we can zoom in to really look at the detail of the evidence. So thank you for listening to my talk. I'm sorry I couldn't be there live. I hope the recording suffices. Hopefully that gives you a little bit of a view into how we create these lists of potential target risks that we show in your report. And if you have any questions, I'm sure that my colleagues will be very happy to answer them. Thank you very much.
Great. So there are a couple of specific questions for Claire, which I think we can probably handle. The first one is, if you disagree with the directionality of compiled evidence or with an element of the evidence, is there a way to change that manually in the platform other than discarding or ignoring the risk overall? And if so, how is that then reflected in the risk scores and in the report? I think, Josh, could you take that one?
I can take that one.
Thank you.
So I think in general, it's a very good question and something that we're actively thinking about. So we want to be able to support not only the rejection of a single piece of evidence or the amending of its directionality, but also the phenotype, the cluster, or potentially the organ system. So it's something we're actively working on. It's not available in the app just yet. grander plans, specifically on like the literature side, is actually annotations where if you say this is the wrong direction and you think it is an opposing one, we'll actually, what we're trying to build is a system where that is flagged internally. We receive that data, we can update your report to reflect that, but also make sure that the models the next week when they're run are aware of that particular incident and improve from there.
Thank you very much. We have another one more specifically about the relationship calculator, which is how do we know this calculator works well and what benchmarks do we use? And I think actually I can answer this question. Um, so we have two internally developed benchmarks that we've manually curated that we use to validate the relationships that Claire was talking about at various levels. One that which we use to validate that the calculator is inferring the correct relationship between a gene and a phenotype or adverse event, given the evidence available, and another that assesses whether, given the multiple relationships per target safety assessment between a gene and its various related adverse events, are these relationships being surfaced with the appropriate relative rankings within a target safety assessment and within a report in our app. So we have these two different benchmarks we've curated, which we keep up to date and test as we have new up to date data coming into the app as well. I hope that answers that question. We have also another question that I think is more directed towards you, Ha, on our literature pipeline. You mentioned PMC and PubMed articles. Do you also process paywalled literature, and how much of the literature that we process is full text?
That's a good question. So the paywalls is something that we have designed to be possible with our technical architecture. So we can process additional sets of literature if that is made available to us. Licensing perspective, that gets a little bit more complicated, but I think we can definitely talk to anyone who's interested in that because we have the capability of doing that processing and tailor make the data that we provide to you so that you get an additional advantage of having a license to additional literature. About the other part of the question, which I already forgot because I answered one question. Can you remind me, Naomi?
Yes, of course. Do we also process paywall literature, and how much of the literature that we do process is full text?
Uh, I think we currently have about 3.5 million full text documents from the PubMed Central commercial subsets, which I think is maybe roughly 50% of all the total literature, maybe a little bit less. The PubMed abstract is a large collection, but I think the PubMed Central is probably a little bit more extensive in terms of what it covers, in terms of all the discussion and results and findings.
Amazing. Thank you very much. And then one final, more general question, um, probably for you, Josh, is what data do we not currently ingest that we think would be useful for target safety assessments?
Oh, that is a never-ending source of fun. There's always going to be data to add to the platform. I think this year we're really focusing in on mechanistic data, so the ability to explore different pathways, whether or not it's a Go process, a wiki pathway, or AOPs. We're currently in the process of ingesting and integrating more single-cell data, so rather than getting bulk tissue expression, a little bit more granular and trying to link those into specific clusters. So key cell types for given adverse events. Um, on the cards, we also have structural information about proteins, so a combination of PDB and AlphaFold doing genome-wide pocket detection, both for interspecies interpretation, but also, what other targets should you be worried about if you're going to interrogate, you're maybe thinking about hitting PDE10, but which other of the PDEs do you have to worry about the most? So those are a few. If anyone has anything in particular they want to be ingested, we're always open to ideas. And ultimately, what we want to make is a platform that people want to use and find useful. And kind of just to echo, you are as well, if you have data that is proprietary to your organization, we are in a place where we can ingest that data within the application itself. So you get a bit more of a bespoke experience. So we know certain organizations are sitting on, I imagine, tera, if not petabytes worth of genetics data, and the ability to integrate that into your report, I think saves us generating a lot of data externally and spending a little less money on experimental stuff.
Great, thank you. I think we will end it there. Thank you everyone for coming and for taking the time. We really appreciate being able to talk to you like this. If there are any questions that come to you after this, please reach out via email or on LinkedIn. We love to chat, particularly about this. And otherwise, thanks for coming again. And please look out for us at some at these events this year. It would be great to talk to you in person. Have a great week. See you soon.
Thanks, guys.